reactions. You remember delta G from your enzyme course. When a delta G, of course, is negative, that means the reaction will proceed spontaneously, like this one here. When it is less than 0, it will proceed spontaneously with loss of energy. It will be exergonic. Delta G is positive, that means the reaction proceeds only if free energy is to be gained. That is like going uphill. So it is this side of the diagram. It is enderogonic. When a delta G is 0, at equilibrium, there is no net change of energy. And in metabolic reactions, we will come across various examples of different reactions that release energy or acquire energy. In metabolism, the exergonic and enderogonic reactions are coupled. Why? To avail energy for the vital processes. Enderogonic processes, of course, cannot proceed independently. For example, a reaction here to go from C to D, from a substrate C to a product D in this enderogonic reaction, it needs to be coupled with another exergonic reaction whereby substrate A is changed to product B with loss of energy. This chemical energy will be captured by the enderogonic reaction and with a surplus amount of energy that will be lost as heat. That is what we mean by coupling of the two types of reactions. Likewise, the catabolic reactions are exergonic, the anabolic reactions are enderogonic, so they are likely to be coupled together. The very famous example here is the first glycolytic reaction, that is phosphorylation of glucose is high enderogonic. So if you have a molecule of glucose, if you want to phosphorylate it using inorganic phosphate to produce glucose 6-phosphate and water, this reaction, delta G, will be plus 13.8. So it is a very high delta G. This will be a highly enderogonic reaction. It cannot proceed under physiologic conditions. So this reaction cannot take place under physiologic conditions. Under physiologic condition, we couple it to another exergonic reaction in which ATP is converted to ADP, hydrolyzed to ATP, and an inorganic phosphate with a delta G of minus 30, highly favorable exergonic reaction. So when they occur simultaneously, we will achieve the final product of having glucose 6-phosphate plus ADP plus water. Methods of coupling, it can be either by an intermediate carrier, for example in the hydrogenation and dehydrogenation reaction, the carrier will carry, or the shuttle, the hydrogen ions between the two processes. Or the coupling could be through a very high energy intermediate like ATP. The biological advantage of having a very high energy intermediate as a form to couple, or a mechanism to couple the exergonic and enderogonic reactions, is that the intermediate is not related to the reactants or the products, and that the intermediate serves as a transducer of energy to a wide range of other enderogonic processes. So it is not specific for this reaction. ATP can be part of thousands or millions of reactions. It is not specific for a certain reaction. High energy compounds are many. The one that you are going to come across as we discuss the various metabolic pathways will be high energy phosphates, at the top of which is nucleosides triphosphates such as ATP, CTP, UTP, and GTP, phosphoenol pyruvate, carbamoyl phosphate, and 2-bisphosphoglycerate, creatine phosphate, not creatinine. creatine with a INE. We have other high energy compounds such as thyroid esters, such as acetyl-CoA, acetylmethylene methionine, UDP glucose, and 5-ribosyl 1-pyrophosphate. We will come across all of these as we discuss nucleotide metabolism, carbohydrate metabolism, amino acid metabolism, lipid metabolism, and pentose phosphate pathway. High energy phosphate is referred to using this symbol. It plays a central role in energy capture and transfer, as we said. It is one of the forms of coupling exergonic and enterogenic reactions. The ATP, as you can see here, is at the middle of the list of high energy compounds. It divides the list into those which are high at the top of the list, including phosphoenol pyruvate, carbonyl phosphate, 1,3-phosphoglycerate, and creatine phosphate. From those at the bottom of the list, which are considered low energy phosphates, such as ADP, pyrophosphate, glucose 1-phosphate, fructose 1,6-phosphate, and so on. So what are the sources of those high energy phosphates? They come from three main sources. Number one is oxidative phosphorylation, which releases free energy from the respiratory chain and oxidation using oxygen. Oxidative phosphorylation from here, from the respiratory chain, ADP is directly phosphorylated to ATP. So respiratory chain is a main source of ATP through oxidative phosphorylation, whereby oxygen, molecular oxygen, is used for the oxidation. Glycolysis, which is breakdown of glucose, also releases two high energy phosphates. And we have the citric acid cycle is a source of one high energy phosphate. I would like you to memorize these sources. The phosphagents as high energy phosphates include creatine phosphate, which is a high energy phosphate of vertebrate, skeletal muscle, heart, spermatozoa, and the brain. While the arginine phosphate is found in invertebrates muscles, they maintain the ATP concentration that is being used for muscular contraction. An increased ATP-ADP ratio, that is to say that ATP is very high compared to ADP, will lead to increase in the concentration of phosphagents. So the body will tend to store high energy phosphate, particularly as a form in the form of creatine phosphate. ATP has two high energy phosphate groups, as you can see here, this one and this one. ATP, adenosine triphosphate, contains two high energy phosphates. They are the ones that are to the periphery of the molecule. On the other hand, ADP, adenosine diphosphate, has two phosphates, but one of them, the one that is most peripheral, is the high energy phosphate. AMP, adenosine monophosphate, of course, does not contain a high energy phosphate. We have what you call the ATP-ADP cycle. It's a very rapid cycle that occurs in seconds. It connects processes that generates high energy phosphate to the processes that use this high energy phosphate. ADP accepts high energy phosphate from compounds above in the list to the ATP, and it donates it to the molecule that is below it. And this is a depiction of the ATP-ADP cycle, whereby ADP takes high energy phosphate from high energy compounds such as succinyl-CoA, pyruvate, phosphoenol pyruvate, 1,3-phosphoglycerate, and oxidative phosphorylation, of course, in the electron transport chain, forming ATP. ATP then donates its high energy phosphate to phosphorylate compounds such as glucose to form glucose 6-phosphate, and also to phosphorylate creatine to form creatine phosphate. ATP, when it acts as a donor to form compounds of lower energy of hydrolysis, the high energy phosphate will be converted to one of low energy. So this is ATP with it is two high energy phosphates. If this ATP is going to phosphorylate glycerol to produce glycerol phosphate, the enzyme will be catalyzed by the glycerol kinase. This phosphate, this high energy phosphate here, will be donated to glycerol to phosphorylate glycerol. However, as you can see, the bond here is a usual bond. It is no longer a high energy phosphate. ATP is usually called, or famously called, the energy currency of the cell. Why is that? Because it is a high energy compound. As we said, it contains two high energy phosphates. It has an intermediate value as a list of high energy molecules. It divides those that have a high energy phosphate from those that are low energy phosphates, and it plays a central role in coupling exergonic to endergonic processes. As an overview of metabolism, what we are going to study. We are going to discuss carbohydrates, proteins, and fats in the step that follows their digestion and absorption. So we are going to talk about carbohydrates as simple sugars, mainly glucose, proteins as amino acids, fats as fatty acids and glycerol. These building blocks or monomers or basic molecules can be catabolized, all of them, to produce a single intermediate that is acetyl-CoA. And why? What is the importance of or the significance of acetyl-CoA? Because it is the first intermediate in what we call citric acid cycle. And what is citric acid cycle? Citric acid cycle is an amphipholic central pathways that links catabolic pathways to anabolic pathways, in which there is production of reducing equivalents that will be used to produce ATP. For example, in glucose, what happens to glucose? Glucose will undergo glycolysis, which is represented by this pathway here. It either in pyruvate or lactate, and this pyruvate will be converted to acetyl-CoA. It will go into the citric acid cycle. We have also what we call BBB, amylose phosphate pathway, that is related to glucose metabolism. We'll talk about it and its role in metabolism. Lipids can also, of course, acetyl-CoA can either go into citric acid cycle or it can be used for fatty acid synthesis, the synthesis of acylglycerols. Here is how the citric acid cycle is related to lipids. We have, of course, we'll discuss lipid synthesis and lipid breakdown. So, fatty acids can be broken down by beta oxidation to produce acetyl-CoA that goes into the citric acid cycle, and acetyl-CoA can use to build up fatty acids. That's just to highlight the central role of acetyl-CoA and citric acid cycle. And this is with regard to protein metabolism. Amino acids we have in our body at any time. We have a pool of amino acids that come from the diet or from tissue protein breakdown and building of tissue protein. We have a central process that's called transamination. We'll talk about it when we come to amino acid metabolism. Amino acids and carbohydrate, particularly glucose, are related because certain amino acids can be used to build up glucose. They have shared carbon skeleton. Amino acids can be broken to acetyl-CoA that goes in citric acid cycle. Amino acids can be broken down to form ketone bodies. Amino acids can of course be deaminated, eventually eliminated from the body as urea. We'll talk about all these processes. And this is just to give you glimpse about how tissues are interrelated or integrated while performing metabolism of a certain biomolecule. For this instance, we are talking about proteins and carbohydrates. What is the relationship of the kidney? Kidney has a role in gluconeogenesis, and it has a role in detoxification of nitrogen products in the form of urea will be lost through urine. The liver, of course, it has a major metabolic role. We have a pathway for urea synthesis, breakdown of storing glucose as glycogen, breaking down of glycogen to give glucose, glycolysis to produce energy. The liver also synthesizes, of course, ketone bodies. It can provide these ketone bodies, for example, to the heart muscle, for example. Here we have an example of muscle and how are they related to metabolism of amino acids. And we have shared cycles between the liver and muscle. We have the glucose alanine cycle. We'll talk about it when we come to carbohydrate metabolism. Don't worry, we'll talk about all of these in detail, inshallah. So we have certain study tips. Of course, you can do whatever you are comfortable with. However, before we start discussing the metabolic pathways, please review the basic courses. So go back and refresh your memory regarding carbohydrate chemistry, protein chemistry, and lipid chemistry, as well as the course in enzymes. In metabolism, we are going to learn concepts. We do have a memorization to do. However, when you approach the subject in form of concepts, it will make memorizing easier. Look at the overall picture and then proceed to details. Be familiar with the nomenclature. It makes big difference. For example, a long name, for example, glycogenolysis. From the name, you can know what is the pathways, what happens, and so on. So the nomenclature is important, and learn how to construct and deconstruct pathways. Deal with it as a play. And then, of course, exercising and discussion with your peers will make the information stick and will make memorization easier and faster. I would like to suggest an approach in how to study a metabolic pathway. First, start by defining the type of metabolic pathway. Is it anabolic pathway? So you will have building up of blocks. You will start from a smaller molecule, ending up with a larger molecules, or is it a catabolic process where you start with a bigger molecule that will be broken down into a smaller one? And what is the role of this pathway? Is it to produce energy? Is it to produce building blocks? Is it intermediate, providing certain intermediates for other pathways, and so on? And what is the basic shape of the pathway? We have linear pathways, for example, like glycolysis, gluconeogenesis. We have circular pathways, for example, like citric acid cycle, and we have pathways that have certain reactions that run in parallel, for example, the pentose phosphate pathway. And memorize what is the initial substrate and the end product. So glycolysis starts with glucose, it ends up with pyruvate or lactate, depending on whether it is aerobic or anaerobic, and so on. And then start to write down or memorize intermediate reactions listing all the substrates, the enzymes, and the products, and then highlight where are the main regulatory steps of this pathway, and then finally, what are the biomedical significance of this pathway? Is it energy production? Is it linked to other pathways? Is there any site of drug action? And of course, if there is any enzyme deficiency that is related to this pathway, it will make a big difference. So if you have any questions or comments, you can share them in your Telegram channel or here in the forum in the Moodle platform. See you in the next lecture, inshallah.